All right, let's jump in here and talk about some cell transport. Okay. Um, so the main thing about transport, all things being equal, things spread out, right? This happens all the time, even in everyday life. If you think about spraying a bunch of like perfume in front of your nose, right, and there's this cloud of molecules there that obviously smell. But someone in the back of the room probably doesn't smell it right away, right? But if you wait a few minutes, they smell it, and that's just because all that cluster of molecules that are highly concentrated right there will start bouncing around and bumping into one another, and they will start spreading out everywhere. And they will eventually get so far apart you won't even be able to smell it anymore. So things like elbow room, they like to spread themselves out. So one reason for this is the second law of thermodynamics. Okay, so says that if entropy of that system will increase unless there's energy added to that system, right? So if you remember the second law of thermodynamics, we can really just sum it up as entropy, right? Chaos, disorder, kind of like this room over here on the slide. Maybe your room looks like that. And there's no way that room's getting cleaned up unless you put energy into it. It'll just keep getting more and more chaotic unless you put energy into that system, okay? So you can have local energy added to a system, right? But remember, globally, you're not going to create or destroy energy, right? But you can definitely do it locally. And obviously, this happens in the universe as well. That's the second picture. So, first types of transport, passive transport. There's going to be a few types of passive. And it's just like what it sounds like. If something's passive, if someone's passive, they don't really have any energy, they're just kind of blah. Okay, so in this case, there's no energy required. It's passive, it's just going to happen. And the reason it happens is because it moves from a high concentration to a low concentration, kind of like the perfume example I gave you. Okay, so diffusion is one of the main types of passive transport. And these are molecules moving across the semi-permeable membrane. And it's pretty much inescapable as long as you can fit. So size has something to do with this, right? The size of the molecule has something to do with whether things can go across or not, okay? So I'm not sure why that's so low down there, the osmosis one. Let's see if I can, well, I think it's just going to be kind of low. But osmosis is really the same thing, but it's really referring to water most of the time, okay, or the solutes, solvents moving across a membrane, okay. So if you look at this diagram right here, this is kind of the typical what we call the U diagram or the U-tube diagram. And so over here you have a lower concentration, right, over here, and then you have a higher concentration over here. And in this case, the solute is a sugar. These green dots are sugar molecules. So there's definitely fewer of them on this side, and there's a lot of them on the right side. And then you can see right here in this box is this dashed line, which is supposed to mean the membrane, right? So in this case, the sugar molecules are too big. They can't fit. So the sugar molecules can't go through, but the water can. So just like this arrow says right here, the water will move through the membrane and come to this side. So notice over here, they're level, but what happens is the water level drops here and raises over here in order to get this side and this side to the same concentration. If you just look at the concentrations on both sides of this diagram now, they're pretty equal, right? So about the same amount of sugar per water on both sides. We're over here when the water is level, there's way more sugar per water on this side than there is this side, okay? And this square just kind of gives you a little close-up of the water being able to go through, but the sugar not being able to go through. So yeah, diffusion is inescapable. This is a pretty basic slide, just another picture, kind of the same thing to show you what's going on. You have some molecules of dye on this side, and obviously there's nothing on the other side. So as long as they can fit through, then things are going to diffuse this way, and we're going to reach what we call equilibrium, okay, or what I like to refer to as dynamic equilibrium. Dynamic means the things will still move. Like the dots aren't just going to get to their side and go, okay, everybody stop. Like that's not going to happen. but then a molecule of dye will go back this way, and then one will go this way. But it's more of a one for one than it is like everybody get to your side and stop, because things constantly kind of move about. Okay, so really dynamic equilibrium is probably a better term. And then down here, they're showing you the same thing with both the orange dots and the purple dots, and as long as they can go across, then you'll have both of them trying to get themselves equal. Tonicity. Okay, so tonicity. We've probably remember talking about hypotonic and hypertonic and isotonic solutions in your regular bio class or your pre-AP class. Everybody talked about it. So the concentration of dissolved substances is lower outside the cell than inside the cell. So the reverse of that would be there's more water outside the cell than inside the cell. So then osmosis causes water to move in, the cell swells, pressure increases, and sometimes it will even burst. Okay. Plant cells, however, won't burst because they have that cell wall there, and so they do become a lot more firm. So a little trick that grocery stores pull off. You've probably been in a grocery store where all the little water misters are going off and they're spraying the produce, right? Well, all of that produce has been, out of the ground or off the vine or whatever for a while. And so, naturally the cells will start losing some water. And then they'll start kind of, shrinking and shriveling, which makes them soft, right? What do people do when they buy produce? They go up and they squeeze it and they want to see if it's firm, right? Because that tells me it's nice and new and fresh. Well, the little trick the grocery stores do is they spray all that produce and then the water goes in to those cells, and then the cells swell a little bit, and then that keeps them with that firmer touch for a bit longer. And so then, they can they're selling fresher produce to people, at least they think they are. So I'll have some pictures here in a minute of these solutions, but for now we'll just talk about them. The concentration of dissolved substances is higher on the outside than the cell, than inside the cell, and so then there would be more water on the inside of the cell than on the outside of the cell, and this is hypertonic. If I back up real quick, I forgot to tell you one little way to remember hypotonic. That's hypo-hippo. Okay, hypotonic solutions, and the cell is going to swell like a hippo because water goes in, the cell swells up like a hippo. Okay. For this one, hypertonic, if you're hyper, you want to get out. Okay. If you're hyper, you definitely want to get out of the house, right? So hypertonic solution, water wants to get out, and water leaves the cell, which means it's going to shrivel, it's going to shrink. Okay. So water moves out, pressure decreases, cells shrivel. This is why plants wilt. When they get in a hypotonic environment, plants are going to wilt and flowers are going to start, tipping over, and so those are kind of just obviously opposites, hypo and hypertonic. So here's a couple pictures for you. I didn't mention isotonic yet, but isotonic is just your equilibrium, basically, the one in the middle where water is. You can tell down here, water is going in and water is coming out. Okay, over here as well, but the size of the arrows and everything are indicating that it kind of comes and goes in equal amounts, and that keeps everything normal. It keeps everything isotonic. Okay, so no net change of cell size there. If we look at hypotonic down here, then we have a lot of water going in, right? We have a lot of water going into the central vacuole right here, and that's going to swell it up. And if you look, the membrane and everything is way pushed out around the edge, and everything's kind of pushed out. And so in a plant cell, it's going to hold because it has a cell wall. In an animal cell, it could actually lyse or split or burst because too much water goes in. It can ultimately burst a red blood cell. And then over here, hypotonic, obviously, water coming out. You can get a very shriveled cell. And then down here in the plant cell, you can see all the organelles come in here to the middle, and they all kind of collapse, and that creates all this space out here between the cell wall and the cell membrane. And so for a plant cell, we call this term plasmolized. Okay, so plasmolized is like what happens in a hypertonic situation. And over here, I didn't mention these terms either. Turgid, you might have heard of turgidity or something being turgid. That's more firm and rigid. Okay, turgid's kind of like rigid. And then flaccid is just normal. Flaccid is just, hunky-dory, everything's fine. So here's just a couple pictures to kind of get you thinking about what things are, right? So let's just go around and I think, why don't you pause the video and try to jot down, like, for every one of these 9 pictures, is it hypo, hyper, or iso? Why don't you pause the video and try to come up with which one is which, hypo, iso, or hyper. And so go ahead, pause it. I'll wait for a minute. All right, you should have paused the video and gone through this. So if we look at just the red blood cells, okay, just the red blood cells for now, and we look at this first one, you have this, or we'll look at all of them. We have this one, and we have this one, and we have this one, okay? So which one looks more normal? Where's the isotonic one? Probably this guy, right? So he's isotonic, and then between this one means water left and one means water went in. Well, this one swelled up like a hippo, so he must be hypo, okay? Probably not going to keep writing on these because it's going to take me forever because I have to do this with a mouse. But that's hypo, and this would be hyper, okay? So I'll just put hyper on that one where water went out. If we look at the plant cells, and we look at what's going on, I always like looking at this one when you see all this stuff collapse into the middle and you see all this kind of stuff out here, what's going on with that guy. Well, same kind of thing. If everything, all the organelles are collapsed in here, that means the water left. That means the water went out, and that one would be hypertonic. And if you look up here, this big yellow guy right here, that is the central vacuole, and he's really big, and he's pushed all the organelles out to the outside now because he's filled up. He's filled up with water that came in. Okay, so that would be hypo. And then down here we have our normal looking plant cell, which would be iso. And then if we go on to just the diagrams, the real giveaway is just the arrows themselves. You have a fatter arrow if there's more water going that way. So if you look down here, you have arrows of the same thickness. So that would be iso. Up here, you have water, more water leaving, and you can tell the cell is actually shriveled a little bit. So that would be hyper. And then up here, you have the bigger arrow going in, and the cell is swelled up. So that would be hypo. So if you haven't figured this out yet, this whole row up here is hypo. Sorry for the H. This whole row here is hyper. And this whole row down here is iso. Okay? All these rows going across are organized that way. So hopefully that'll help you out. Just another kind of couple questions, little example. So which way will the sucrose go and what if it can't? So in the beaker or in the environment, okay, which in this case is going to be this beaker, we have a 0.01 molar sucrose, a 0.01 molar glucose, and a 0.01 molar fructose. And then in the cell, we have a 0.02 molar glucose and a 0.03 molar sucrose. So which way will the sucrose go, if it can? So if it can, will the sucrose leave the cell or will sucrose come into the cell? Well, if it goes from high concentration to low concentration, which one has more concentration? This one does. So it wants to go out. And we don't know from this diagram whether it can or not, but it wants to come out. And then the cell would shrivel a little bit. If it can't do that, then water would probably, go in its place and try and equal out all these concentrations, okay, of these sugars. This is kind of an interesting diagram because of the, most people just kind of think about fish being fish and not really realizing they have to regulate themselves differently. So it depends on if you are a marine fish out in the ocean or if you are a freshwater fish in a lake or a pond somewhere. And so how do they go about doing this? Well, let's look at the arrows again of what's going in. So this guy's out, he's a marine fish, he's out in the ocean, right? So when he takes in water, he's getting a lot of salt, right? He's getting a lot of salt ions in there. He's also getting it from his food, he's getting it from the drinking water, and it's coming in, okay? So the dark blue arrow is the salt and the light blue arrow is the water down here. So we have things coming in, and as they go across the gills, he's getting rid of a lot of salt, right? Excretion of salt ions from gills, and over here, osmotic water loss through gills and other parts of the body surface. So he's given up fair amount of water as well. And then down here with his excretion, excretion of salt ions and small amounts of water in scanty urine from kidneys. So they can afford to get rid of a lot of salt, right? Because they have so much salt in the ocean, he doesn't really need a bunch. But he tries to hold on to a decent amount of water. That's why you have a really skinny amount of fresh water coming out in the urine, because it's trying to hold on to as much of that fresh water as it can. If you look over here in the freshwater fish, clearly has a lot of fresh water, but doesn't have a lot of salt. And so the only way to really get salt or some of those ions is through its food. And so it takes some in, right? But the gills try to take the salt out. So they uptake the salt. Okay, they want to take the salt out of that that water or that food if they can, and hold on to it. And they're going to get, look at the size of the arrow over here. They're going to get rid of a lot of water, a lot of fresh water, because they live in it. They have plenty of it. And so, but the little tiny arrow next to it is the salt. They're going to try and hang on to that salt and not get rid of very much. So pretty interesting, different osmoregulation depending on what type of fish you are. So we have here a contractile vacuole. I think we've talked about this before. Paramecium are single-celled protists, and they definitely live in aquatic environments. And this contractile vacuole, this star-shaped vacuole, kind of spreads out like your hand, if you remember, and the water will fill up. Because they live in water, water is constantly coming in, so then this thing will constantly try to squeeze out a bunch of the water to maintain the type of environment that it's in, to help them survive a hypotonic environment. That contractile vacuole has to constantly do this, okay? All right, a little bit more on passive transport here. So diffusion fit in there, osmosis fit in there, and now we're going to talk about facilitated diffusion, also passive, okay? So no energy required. So they do require something else, though. They require the help of transport proteins, okay? You have to have transport proteins when you do facilitated diffusion. You have to have these guys, okay? So they also might have aquaporins, which are big channels for water to go through, for moving bulk water, for moving a lot of water at once. And sometimes there's ion channels as well. So if we look at a couple things here, grab this. So this is your channel protein. This like purple guy right here is your channel protein. So that is that's one of the transport proteins. And the other one looks like this, the carrier protein over here. Okay, so you have your membrane. And really the way I try to explain this is clearly there's more big red dots on this side than there is this side. So they want to go down, right? They want to go across that membrane. Now, if you have like a chain link fence around your backyard, can everything go through that fence? Well, some things can, right? Small birds and squirrels jump through there, and the air goes through there, and insects go through there, butterflies and bees go through there. But your lawnmower doesn't go through there, right? You can't just walk it through the fence unless you open the gate, right? The gate allows you to get something bigger through the fence, and that's what these transport proteins do. Okay, they're kind of the gates. They're kind of the tunnels or the gates to get something from one side to the other. For bigger objects. This is strictly like a size thing, okay? So the channel protein, just like what it sounds like, a channel, a tunnel. It just kind of sits here like this and things pass through. Over here, you have a carrier protein, and you can see right here, they kind of connect on the one side, and then over here, they do this. And so what really this is a timeline, okay? So they're trying to show you what would happen over time. Okay, so you have all the big red molecules up here. So they want to go this way. So the big red molecule comes in here, it binds to the carrier protein, right? And when it binds, it causes a conformational change, and it flips to this configuration now, and then it's able to be released to this side. So that carrier protein just sits here and toggles like this, okay? So it sits open, and if something binds to it in that space, then it's going to flip, and then it's going to let it go to the other side. And once that leaves, what do you think happens? Then it flips back, okay? So that's kind of how carrier proteins work. You have to have these proteins for passive transport. But again, it's still high concentration to low, no energy required. Now we move to active transport, and just like what it sounds like, it's active. It's going to require energy, right? So way I kind of explain it is passive transport. If you're sitting in a canoe and you're not paddling, you're just going to float downriver, right? You're going to go whatever way the current's going. If you want to go the other way, if you want to go upstream, what do you have to do? You have to paddle. You have to put in energy. Okay. So these are going to go upstream. These are going to go against the concentration gradient. They're going to go from low concentration to high concentration. They're going against the current. So it's going to require some energy. So you can see in this first picture here, you can see we have very few hydrogens here, and we have a lot more over here, but we're still going to send them through a transport protein, also required, and we're going to do that spending some ATP to do that, okay? So substances always go from low to high when you're talking about active transport. It's the only one that goes low to high, active transport, okay? So another kind of typical picture here, and again, it's a timeline. So this whole thing is, whoa, that's a horrible line, but that whole thing is the timeline, right? We're going this way across. There we go, a little better. Here's your timeline going this way. So we have just a few big molecules on this side, whole lot more down here. And so the ion comes in here, binds with the carrier protein. Okay, that's going on right here. And then eventually you put in your ATP, you put in your energy, and it's going to flip the carrier, and then it's going to be released to the other side against the concentration gradient, and it's going to go from low to high. So that's kind of the big deal about it. And phosphorylation has to do with the ATP there. The energy put into the system, which is adenosine triphosphate, right, is responsible for the conformational change of the protein. And so phosphorylation, we're going to get to this down the road. Phosphorylation is going to, you're going to come to know it as a trigger. Whenever something gets phosphorylated, something's going to happen, okay? And so you break the ATP, and so then you have this free phosphate, and that phosphate attaches to a protein, and a lot of times it causes a conformational change. It causes a shape change or something else. So we're going to talk about phosphorylation a lot more down the road. And another example here of active transport, probably the more famous example, is the sodium-potassium pump. This is what makes your nervous system work, okay? This is how we can send action potentials up and down our neurons so that we can do things. So if you look here at number one, you're at the cytoplasm, and you, at the beginning, the sodium is low on the inside, but the potassium is high, and obviously the reverse is true on the outside, okay, where the potassium is low and the sodium is high. So the sodium is low here, so the sodium is going to come in here, and it's going to fill this slot. And when that third slot gets filled, and we spend some ATP, right, and by spending it, I mean we break it, we break the bond. And so you can see here, when you break adenosine triphosphate, then you get adenosine diphosphate plus this free one right here. So this free phosphate is going to phosphorylate this protein or cause it to change shape. And so you have these three sodiums. And then when it comes down here, this thing phosphorylated it. So now the protein opened up to the other side, and the three sodiums all went this way against their gradient. Because look, sodium is low to high. So they all went up and out against their gradient. And then we have, since it's open to this side, and you see these two active sites over here on this side, and they fit potassium, right? They have a different shape. They fit potassium, okay? Then when they come in, that's actually going to kick the phosphate back out. That's going to remove that phosphate. Well, if that phosphate gets kicked out, what shape will the transport protein now take on? Its original shape, and it will open back to this side, and then your potassiums are going to come this way. And again, oh goodness, and then again, your potassium will go from low to high. So we send 3 sodiums out, and we bring 2 potassiums back in. That is the sodium-potassium pump. And later on, we'll link this a little bit better to the nervous system. But this system right here is what allows your nervous system to work. If you couldn't do this, then you wouldn't be able to have any action potentials. You wouldn't be able to have any neurons fire and carry any messages anywhere. So this is super important. lot of your ATP that gets generated in the mitochondria are spent on the nervous system doing this. And let's see where I can put myself here. Let's go there. So active transport, we have this co-transport here. So basically what's happening is you're going to have the original active transport, okay? You're going to have the original part right here where you have your hydrogen, you have your ATP, and we're pumping protons out here, and we have all these protons out here now, low to high. But what happens is we have so many out here now, by themselves, what would they want to do? Well, there's so much out here, they want to go back in, the hydrogens do. And actually, sometimes you have to bring in other things like sucrose. And in this case, this is a sucrose proton co-transporter where you have to have something here and here in order for it to bring these things in. So the hydrogen then will come in here and dock, and then the sucrose comes in here and it will dock in its active site, and then they both get brought in together. There will be a conformational change and the protein will open up and bring them both in. So co-transport, working together to bring a couple things in. And then now we're kind of wrapping a few things up here. We also have bulk transport, what I like to call Costco transport, okay, like the 50 rolls of toilet paper, okay? So endocytosis, there's 3 of these. Phagocytosis is cell eating. Penocytosis is cell drinking, which are just small solutes. And then receptor-mediated endocytosis is just the cell being picky, okay? So you have in the first one phagocytosis, bigger solid particle, bring it in, merge with the membrane, and bring it into the cell because it's endocytosis, you're bringing things in. Penocytosis, just little tiny things, little solutes, little guys, so we call that cell drinking. And then receptor-mediated, you can see on this last one over here, you can see these receptors right here. And the receptors are there, and they have to fit whatever is coming in. Only some of these things are going to be able to come in because they have to be able to merge with this receptor. They have to fit like puzzle pieces in order to get them in. So this is the cell being picky. It's kind of like having to have the password or the key to get in. You have to be able to fit. You have to be able to match up. So that's structure and function again. And then we have exocytosis, which we release a large amount of materials outside the cell, and it's down here. It's down here, right, with when you're getting rid of a bunch of things all at once. And so you have some exocytosis there, and this picture is very similar to the one, it says remember me over here, very similar to the picture of the 4 fates of the vesicle, right? And so that was definitely one of the fates of the vesicles, was that things get exocytosed outside of the cell. Okay, so bulk transport, kicking out a lot of things out of the cell for exocytosis. And we made it. So there's some practice questions for you, and hopefully that'll help you guys out. Talk to you later.